Sorry to interrupt, but the webinar is
so what we say now will be recorded.
Yes.
Okay, you're opening up, are you, Jiang?
Yes. Okay.
Okay.
Yeah, people are. People are getting in.
Okay, great. Well, welcome everyone. We've just opened up. Plenty more participants should be coming, so we'll give them a few minutes for sure to join. But welcome, please stay online and we'll get started soon. Thanks. Okay, I see the number of participants joining is slowing down, so I think we've got the majority of those who'll be attending today already in, so I'll get started. Welcome all. My name is Oliver Wootton from UNITAR, the Chemicals and Waste Management Programme. Thanks very much for joining us on the latest of our webinar series, so particularly this series we've done in collaboration with the Secretariat of the BRS Conventions. For UNITAR, we're very glad to continue our work on nanomaterials and nanotechnologies. We have been looking into the links between nanomaterials and plastics of late, and today we have a webinar on micro nanoplastics and the marine environment. And we have a few panelists who will be presenting. So we have Tony Walker, who will come in a minute. We have Bart Koelmans as well, and then we have Mehrnoosh Azodi from the BRS Secretariat. We will then have a question and answers session. And then my colleague, Georg KARLAGANIS from UNITAR will give some concluding remarks. As per usual, we will share the presentations once we have agreement from the speakers and a recording so that people can access the information. And of course, to those of you who may be interested, we have our e-learning course online on UNITAR's page and various other materials related to nanomaterials and other chemicals and waste management issues. So please do have a look around our website. The BRS website has information as well. But with that, I will pass over to Tony. Please introduce yourself and then please go ahead with your presentation. Thank you.
Okay, so I'm actually doing the sharing and introducing at the same time. So yeah, as Oliver's already mentioned, I'm Tony Walker. I'm a full professor in the School for Resource and Environmental Studies, which is based at Dalhousie University in Halifax, Nova Scotia, on the east coast of Canada. So I'm going to dive right in because we only have 25 minutes. Well, I do. And also, I'm just going to give a recap of some historical research in terms of microplastics in the marine environment, but also some contemporary research as well. So let's dive in. I know I'm speaking to the choir here, and I really don't know how familiar many of the audience members are with the size class definitions, but generally, for the purposes of this talk, I'm really going to talk about particles, plastic particles that are less than 5 millimeters in dimensions. And in terms of the different types of microplastics, well, that's a webinar in and of itself. But here on the bottom left, we can see some fragmented macroplastics breaking down into smaller microplastics, and they're often referred to as secondary microplastics, degraded macroplastics, as I'm sure you're more than familiar, as well as primary microplastics, which were intentionally made and manufactured for beauty and cosmetic or healthcare products. In some countries, they're banned. We'll talk about that later. And also a type of microplastic that does keep me awake at night is microfiber plastics, the stuff that really sheds from our clothing during the life of the wearing the materials, manufacture of the materials, and also end of life and washing. But that's just the quick 101 on the types of microplastics. But you know, the term microplastics has only been around for just over 20 years now. And if we look to this seminal paper by Richard Thompson and colleagues back in 2004, it seems so recent, doesn't it? That was the first time that the term microplastics had been recorded. And it was a case back then of, finding it in different environmental compartments, and wherever you looked, we found it. But what we've learned in the intervening 20 years, and there's a subsequent paper here shown on the right, also with Bart Koelmans as a co-author, we've learned a lot more about the fate and transport and effects of microplastics, and we'll hear more about that from Bart in the second presentation. But even amongst the academy, there's a wide debate on that size classification. And as you can see here, that not all researchers are using the same size classification, but for the purposes of this talk, I'm going to be talking about microplastics that are less than 5 millimeters in the longest dimension, down to 1 micron, or 1 nanometer if we're talking about nanotechnology. And here you can see Bart and colleagues is also consistent with that upper limit here from those studies. But you know, the full disclosure here, and just to dispel any misconceptions, that microplastic pollution is not just pollution in the marine environment. Again, speaking to the choir, I'm sure you all know, but microplastics now exist in every single environmental compartment on Earth, including the atmosphere, the air we breathe, the upper atmosphere, soil, obviously marine, in ourselves. And there'll probably be other webinars on that topic, as well as aquatic and terrestrial biota, and in freshwater ecosystems. But as you know, the title of this presentation is Microplastics in the Marine Environment. So we're going to focus mainly on those studies, including my own and from my lab, as well as others. But in order to understand the emergence and the timeline, the chronology of microplastics in the marine environment, we need to take a step back. And also just imagine where all these potential sources are coming from. So this is an interesting infographic. It's quite busy but informative, from produced by Grid Arundel in collaboration with the United Nations Environment Programme. And as you can see, any anthropogenic activity that we do, we interact with plastics, and those plastics, as we've talked about, degrade into microplastics. And so if we take a deeper dive, we can look at degradation from physical processes, sun, UV radiation, transported by wind across the Earth atmosphere, road markings, paint, tire wear particles, artificial turf. I'm sure that's not a huge contributor in terms of the global budget, but it's just to recognize that anything really made of plastic polymers does degrade over time, and that's just one example. These are land-based sources, obviously. I've mentioned the primary microplastics that were intentionally made in cosmetics and beauty products. Clothing and washing, so shedding of those fibers, which we talked about, industrial activity, just manufacture of the plastic pellets themselves and accidental leakage into the environment. Wastewater treatment plants is another point source into the marine environment, as is agricultural sector, which is absolutely massive, and it's really probably comparable to other the kind of budgets that we've been thinking about in the marine environment. Plastics in the terrestrial ecosystem from agriculture is just as big. And then, of course, we get down to marine sources from fishing activities, the shedding of rope, the shedding of nets, and also I've noticed some colleagues here on the call abandoned, lost and discarded fishing gear. And then subsequent degradation of macroplastics, which we've also talked about, and paint from ships, the grey water and black water from ships, and also the aquaculture industry. I'm going to actually link back to some of that later on. And these are the marine sources. So the term microplastics has been around for a little over 20 years. But we've actually recognized that there's tiny plastic particles polluting the marine environment well over 50 years ago. So if we look back to a paper by Thor Heyerdahl during the expeditions on the papyrus raft ships across, not ships, but boats, across the Atlantic, both in 1969 and 1970. So this is well over 50 years ago. Observations were made of these tiny plastic particles in the size range that we've been talking about. So they've been recognized, but in terms of the extent and magnitude of this pollution, it was still very much in the early days of microplastics research. Just a couple of years later, the work by Carpenter and Smith, primarily looking for, plankton research, ended up uncovering tiny plastic particles, again in the Sargasso Sea, in high concentrations near the sea surface, and again in the size ranges that we were talking about earlier on when we were defining them. And here they are. They comprised of pellets back in the day, and also those fragmented larger macroplastics that were at the sea surface. And a few years later, other researchers had, so that it wasn't just confined to the Pacific Ocean, the Atlantic. They were found in the Pacific Ocean. And this study, and published in Nature in 1974, again, similar size classification, researchers primarily looking for phytoplankton samples, were also inadvertently collecting microplastics, but they didn't term it microplastics at that time. And it wasn't until Charles Moore and colleagues in 2001 concentrating their research efforts in locations in the North Pacific Gyre, they really started to find incredibly high concentrations, much higher than in other regions of the world's oceans. And so, of course, again, size classifications were consistent with what we've already discussed and previous research. Anything less than 5 millimeters in diameter. But these were found in much higher concentrations than had previously been reported in these gyres. And of course, that brings us to the infamous ocean garbage patches, which we all recognize. We're all familiar with the terms and the terminology. But here are the 5 main gyres on Earth. And of course, here the concentrations of microplastics are much higher than elsewhere. But another misconception that I'd like to dispel here is that once in the marine environment, microplastics don't just exist at the sea surface. Obviously, there's different buoyancies with different plastic types, which I'm sure many people on the call already know. But they don't just exist at the sea surface. They exist in other compartments because we know that from our research. And here's a paper from Bart. Sorry to preempt this paper, Bart. You may have it in your presentation later. But Bart and colleagues actually estimated with mass budgets that roughly 1% of the microplastics or the plastic budget existed at the sea surface, with the majority actually being transported down to marine sediments, deep, shallow, coastal. But they rapidly sank through different processes, which I will talk about later, but accumulating in the sediments with less than 1% or about 1% at the sea surface. So with 1% at the sea surface and, hypothesized in a mass budget, ending up in the marine sediments, is that the final fate of these microplastics? Well, that remains to be seen. In fact, microplastics continually move around the environment, the marine environment, especially for the purposes of this talk. And in fact, once in the environment, they're transferred from the sea to the atmosphere through processes such as storm events and wave spray energy. And once into the atmosphere, they can be redeposited back into the marine environment, or depending on trajectory and wind speed and wind direction, back onto land-based ecosystems. And so really, microplastics, once in the marine environment, move around pretty much like hydrological cycles or the carbon and nitrogen cycle. So they're in constant flux. And it seems, as far as we know right now, we're finding them in all kinds of compartments, but they don't necessarily stay there forever. And one way to test that hypothesis was with some of our own research. And here you can see the North Atlantic garbage patch with the high concentration or the relative concentration of small particles in the North Atlantic. And we had a study with a couple of grad students a few years ago during Hurricane Larry. So for those in North America, especially Canada, you'd probably recognize that name. But Hurricane Larry was unusual when compared with the tracks of the previous hurricanes since the 1850s. This one didn't actually make landfall at all. It actually only passed through the ocean, the North Atlantic garbage patch, and the only landfall it made was in a remote area of Newfoundland and Labrador, where we had students deployed as we tracked the storm. And they measured the deposition of microplastics in this remote environment, far away from large urban centers. And with robust glass jars and tenacity, these grad students measured passively rainwater fallout both before the storm, during the storm, and also after the storm had passed. And these are 6-hour intervals of passive rainwater collection. And even before the storm made landfall, we were measuring somewhere between 30,000 microplastic particles per square meter per day. And at the peak of the storm, we measured a whopping 113,000 microplastics per meter square per day in a remote environment where there was no large urban centers. So all this was sourced from the marine environment and from the garbage patch. And many of those particles are in the 2 to 10 micron size class. That's important. Please remember that, because I'm going to talk about that next. And those tiny particles, whether they exist at the sea surface or they do finally get deposited into the sediments, that's not their only final fate. they are also incorporated throughout the food web, right from primary producers at the bottom of the food web, through zooplankton and through their subsequent predators, right up to the top of the food chain, including ourselves. And they're not just ingested passively into the gut and then passed out, as we'll see through these other local studies with myself and my grad students. We'll see that they're also internalized. So in terms of that transport to the marine sediments and the fact that microplastics are interacting at all trophic levels, and again through atmospheric deposition, a paper here by Melanie Bergman and colleagues recently found high microplastic concentrations, much higher than ambient seawater, trapped inside primary producing sea algae. And so these microplastics were being entrained and deposited from the atmosphere into Arctic sea ice, into Arctic snow. And as the sea ice algae attaches to the bottom of the ice flows and start photosynthesizing, they were incorporating microplastics. This changes their buoyancy, and during decomposition and settling, they migrate to the sediments. Those primary producers were also harvested by ingestion through predators, and then also transferred through the Arctic food web. And these were the tiny particles, but research I'd done in the past had measured microplastics in commercial fish, and that was a really kind of like a red flag at the time. Because we found out that many of these were commercially important species of fish that were consumed by humans. But at the time, it was thought that these microplastics only existed in the gastrointestinal tract of these fish. And in this study, we measured 11 species, and 9 of them all contained microplastics. But these were the larger size classes. 2.5 millimeters, so fairly large. You could probably still see them with the naked eye. We measured the different polymer types, but it was believed that these were actually passing through the gut, and so the edible tissues remained safe to eat. However, that wasn't the case. This is the elephant in the room that I'd like to talk about now. Is here in Nova Scotia. This is the province of Nova Scotia in eastern Canada. The biggest economic driver here in this province is the commercial lobster fishery. Many of those lobsters are eaten domestically, but also exported. And unlike the microplastics that we'd found in the past in the gastrointestinal tract of fish and shellfish, including lobster, what we aim to look at here was the smaller microplastics in the fish tissue, i.e. the tissue in the tail that was consumed by humans. So that is the elephant in the room. And what we found, every single lobster that we sampled throughout Nova Scotia contained at least one microplastic particle. And in some cases, they were in much higher concentrations in the most intensively fished areas of the province. In this case, southwest Nova Scotia, where the fishing effort and the fishing intensity was greatest. And we opined and hypothesized that this could be rope fragments and also the actual lobster traps themselves. So we also need to go back and conduct more research to do some matching of the forensic of the plastics used in the fishing industry with the plastics we found in the fish tissue. And again, many of these were of the size classes that I mentioned earlier, the really small size classes, less than 10 microns, and the majority that we found in the lobster tail tissue were 2 to 3 microns. So these are able to now pass through cellular tissue, both in fish and shellfish, and also other organisms, which we'll, if I have time, we'll get to look at next. Another important seafood product here in Nova Scotia and eastern Canada is aquaculture of bivalves, in this case blue mussels and oysters. and they're filter feeders. And so what they are fantastic at doing is filtering at least 30 times their body volume per day. But by doing so, in order to filter feed, they also inadvertently entrain microplastic particles. The physiology of these bivalves is pretty good because they're able to also eject them when they recognize a foreign particle. But what we wanted to see is if there was a difference here in the retention of microplastics between the two species. So we looked at different sites around Nova Scotia where mussels and oysters were co-located. And what we found, microplastics in all of these individuals, yet again, no surprises. But what we did find were the blue mussels were retaining higher concentrations of microplastics than oysters. And we believe this is a function of their differences in physiology and their ability of oysters to ingest microplastics, but they still contain microplastics nonetheless. And it doesn't, you know, the entire food web is impacted as far as we know. Every time we look at different organisms in the marine environment, as well as the terrestrial environment, we're finding microplastics in different tissues. And this was a study conducted by colleagues looking at a long-term data set, looking at different species of marine mammals, collecting different tissues. And they found microplastics in pretty much 2/3 of all tissue samples they found in these multiple species over 20 years. And what they found that these microplastics, tiny particles, were translocating primarily to blubber. And it's believed that plastic particles, microplastics, are lipophilic, which means they do adhere to fatty tissue. And in this case, that was measured and determined in whale blubber and seal blubber. But what kind of solutions do we have to actually turn the tide on microplastic pollution? There's clear, overwhelming evidence. But what we find is, again, we're on a United Nations related call right now. And so one of the exciting things that's been ongoing since March of 2022 is the ongoing Global Plastics Treaty negotiations, which still hasn't been finalized. It's still highly contended by petroleum and plastic producing countries. But the important thing here is to recognize that most of the solutions put forward in the different iterations of the draft so far are primarily targeting macroplastics. And that makes sense because we know that macroplastics are a source of those secondary microplastics. So turning off the tap is an obvious solution and widely recognized, but it really is still the elephant in the room when you factor in all those other types of microplastics in the environment. And so myself and colleagues, we believe that capping and having limits on primary polymer production is key to move forward, because we certainly have to close the tap before we can start to develop solutions to attack these legacy pollutants in the environment. So capping is obviously a solution. And also there's a myriad of other a patchwork of solutions across the world. United Nations, even before the Plastics Treaty, had solutions to tackle plastics and microplastics. And there are all kinds of instruments around the world. But we really do need a single harmonized solution that's standardized. But what we see in terms of solutions to address microplastics specifically, there's very few international or regional regulations. Examples here include California, a state in the US, which is to my knowledge, one of the first, if not the first jurisdiction in the world to regulate microplastics in drinking water. And then there are other regulations in both in the US and in Canada to ban microbeads in cosmetics, and that occurred a few years ago. But as you can see here, the map at the bottom really shows where there's legislation to tackle microbeads. Yes, it's important that there's legislation that exists, but we still have a long way to go. And I think that takes me to the end of my presentation. Thank you.
Wonderful. Thank you very much, Tony, for that very interesting information, very sort of deep scientific information, and then linking it to the UN Plastics Treaty negotiations. Very interesting. Bart, I will then pass over to you. So please introduce yourself as well, and then go ahead and share. Thank you.
Thank you, Oliver. All right, so thanks for the kind invitation to present here about risk assessment of microplastics. My name is Bart Koelmans. I am heading the Aquatic Ecology and Water Quality Group of Wageningen University in the Netherlands. And I think over the past 15 years, I've been working on detection of micro and nanoplastics and fate of the particles in aquatic environments mainly, but also focusing on the effects and the risks. And from there, I've also been involved in policy advice for the World Health Organization and the European Commission and several other bodies. Yeah, I think this talk fits nicely to the one just presented by Tony Walker, in the sense that he explained about the definitions and the abundance of microplastics in the marine environment, and the various forms and shapes the particles can have, and also the translocation into the bodies of organisms, right? So the whale and the lobster and fish and shellfish. So on many occasions, the question is asked, this exposure, does that yield, does that result in effects, not only in the lab, but also in the natural environment? Now, that's quite a difficult question to answer, but I will try to do as good as possible. And first explain about what you would need in essence for a good and consistent risk assessment. And then you need 4 things, like in a pizza quattro stagioni. So the first 2 things you need are an exposure assessment and an effect assessment. So the exposure is then about what are the concentrations of particles. And the effect assessment would be something like at which concentration of particles does an effect start to occur. And then if your concentration, the exposure is higher than that effect threshold, then you could say there is a risk. If it's lower, there's no risk. Now, in a very simplistic way, this is how it works. There is an important thing, and that is that the exposure should be expressed in a unit and metric, which is exactly the same as the effect assessment. So if you are talking about numbers of particles, let's say from 1 micron to 5,000 micron, which is an often used definition of microplastics, then the effect assessment, so the effect threshold, should also be expressed in that same number, concentration, and unit. So I'm not saying that this is the actual relevant metric, but at least as an example that these 2 should be aligned, because only then you have an consistent and meaningful risk characterization. Now, for traditional chemicals, this is something like, milligram per liter for organic compounds or heavy metals. But for microplastic, it's far more complex because the material is so diverse. And then the 4th thing is that if this is all done properly, there's an important component, and that's how do you communicate the risk and how do you manage the risk? For instance, by some of the mitigation methods that Tony Walker also alluded on, and hopefully can be enforced by the Plastic Treaty. All right, another way to look at it is via this scheme, which starts with saying that you need to define the problem. So for what part of the ecosystem, for which population or which community are you assessing the risk? And then you would go to the middle block here, where you would have to think about what do you think the effect mechanism is? And if you have an idea about the effect mechanism, then what is the metric that you would choose to characterize the exposure and the effect, like I explained with the previous slide. And the metric that you would choose, thinking from the effect mechanism, can be referred to as the ecologically relevant metric, the ERM. And the yellow arrow points to several candidates for that metric, like mass, surface area, volume, or aspect ratio. For each of these, there are some hypothesized mechanisms where that would be the key metric. And from here, you could already say, well, maybe it's not just one of them. Maybe some of those are important at the same time, because the particles may cause effects through different mechanisms that occur simultaneously. And then in the last block, it is about aligning those metrics and doing this comparison of exposure and effect threshold to, at the end of the day, make a conclusion about risk, to answer the so what question. Does the presence of these particles matter? How do we know that some of those metrics are more important than others? And I give 3 examples here, but there's far more literature, but I would say these are maybe some of the key studies that are done. There was a weight of evidence analysis done where the authors analyzed 105 studies. And here we basically checked how many times authors would say a certain mechanism is suggested or demonstrated. And also a quality analysis of the data was done. And then it came out that food dilution and oxidative stress were the most mentioned mechanisms for which there was the highest evidence. Now, food dilution, what type of a mechanism is that? Well, it's mostly important for small organisms like zooplankton species. You can imagine they normally feed maybe on phytoplankton or very small organic particles. And then if they would eat suddenly microplastic particles that have a low nutritional value, then you can imagine that basically the value of the food, the nutritional value, is diluted. It's diluted by this inert material. So this is referred to as the food dilution effect, and it's especially important for small organisms, logically, as long as we talk about microplastic. Another one is oxidative stress or inflammations, for which it's thought that the surface area of the particles is important. It's about the biological incompatibility of the polymer surface, perhaps with certain functional groups, with the biological material in an organism that cause oxidative stress or inflammation, dependent on the shape also of the particle. Now, if it's food dilution and oxidative stress, well, for dilution based on volume, you would need the volume as the environmentally relevant metric. And for oxidative stress, coming from the surface of the particles, it would be the surface area. Another study just tested 160 studies and correlated the effects observed in the lab with certain aspects of the particles the animals in those tests were exposed to. And then there were 2 aspects of the dose, 2 aspects of the particles that turned out to be significant. And these were the only 2. It was the surface area, p-value is indicated here, and volume. So this was consistent with the weight of evidence approach. And there is a 3rd study in preparation where machine learning modeling is used with a lot of data points, where also the surface area and the volume came out as the best dose descriptors. So this is still hypothesized, but altogether it represents quite a strong body of evidence that these are 2 relevant effect mechanisms to take into account for a risk assessment. And it means you would do the assessment for the mechanism of food dilution and for the mechanism of oxidative stress. Are these 2 additional? Can you just add the effects? No, because it's different mechanisms and we would not know how to add the effects. So that means you would just do the risk assessment for the one and then the other, report that to, for instance, policymakers, and then they could, for instance, decide that one of the 2 gives a higher indication of risk, and then using the precautionary principle, you could focus on that one for any policy measures. So far, so good. Another thing to consider when assessing risk is that not all particles fit into all organisms. plastic items can be considered as a continuum. No plastic items, no two plastic items or particles are the same. Two microplastic particles are always very different, but they come in very high numbers. And this means that if you think about the characteristics, you can consider them as a continuum, as you can see here in the middle of this figure. Now, obviously, the larger animals here, including humans, well, they could ingest those larger items, could suffer from them. However, the smallest particles here at the bottom of the graph, yeah, they can only be exposed to the small ones, because otherwise it would simply not be ingestible. So bioavailability, ingestibility must be taken into account if you assess risk, because for the smallest organisms, the larger particles are simply not bioavailable, so they will also not cause a risk. Then there are two challenges that I would like to explain. And the first one is about the fact that not all data that are reported in the literature, not all effect data, for instance, or exposure data, are equally fit for the purpose of risk assessment. That doesn't mean that this data or these studies are not valuable, not at all. They're always valuable. Authors do the studies for various reasons, and conclusions are consistent with the study aims. But risk assessment is a specific thing to do with data, and then not all data are equally fit for the purpose of that. So this is from a study that looked into the appropriateness to use certain data that could be used in a regulatory context for risk assessment, for instance. And data sets can score to a maximum of 2. And then you see that certain aspects of studies, like for instance, indicating where the microplastic that has been tested comes from, scores quite well. However, the verification of contamination of the system scores very low. So certain criteria that studies should fulfill, ideally, are well represented and well fulfilled, whereas others are less well fulfilled. And it means that not all data are equally fit for purpose. And that's important to take into account. Another challenge is that the data that we measure and the data that we get from effect tests are not really aligned. For instance, somebody could use a method, and the method would capture this part of the microplastic continuum. Whereas in the laboratory, a test could be done with maybe 10 micron polystyrene, the spheres here, left-hand side lower corner. It's both microplastic, but it's completely different. So if you would try to align the exposure in the rectangle with the effect obtained for the monodispersed polystyrene particles, it would be an apples-orange comparison. So an alignment is needed. Now here at the left-hand side, you see a graph which illustrates how diverse the targeted size ranges are that researchers have used in their studies. And it's logical. There's a certain method. You see all those studies here at the left-hand side listed. And the bars that you see indicate the size range. So on the x-axis, it's the size range from 1 to 5,000 micrometer. And those arrows indicate which size ranges were targeted. And it's very diverse. Now, this can be aligned in the following way. Imagine that the abundance of particles on the vertical axis here at the right-hand side, so the relative abundance, if you plot that as a function of the size of the particles on a log scale, then often a more or less straight line is observed. That is a power law or Pareto distribution, and this is often observed in the environment, also in marine environment. And imagine that you only measured the range in this yellow parenthesis, right? Then if you know the distribution, if you know the mathematical equation for that, you can realign that number concentration into what you would have measured if you would have if your method would have been able to target the entire microplastic size range from 1 to 5,000 microplastic. So that's mathematics. Some equations are involved. It is not perfect, but it's better than to compare those unaligned data sets. So this makes it all more comparable. This has been applied in actual risk assessments. This is an example of an alignment of these of exposure data. So this is a cumulative frequency distribution for microplastic particles in surface water. You see the concentrations on the x-axis from very low to 1,000 particles per liter. And you see this curve with black data points. And these are the unaligned data, the original data from a lot of studies. And then the data were aligned and we got the blue curve, the blue cumulative frequency distribution. Now the alignment makes the concentration higher because the original methods would have missed a part of this microplastic continuum. So the aligned data are higher. And then just to make the point that this can be used for risk assessment, we plotted a critical effect threshold here at 90 or 80 particles per liter. That's the vertical orange line. And from this, you can conclude that a certain percentage of the surface water concentrations would have a higher concentration than what could be considered safe based on laboratory results for effects for aquatic organisms. So this is alignment for exposure data. There is another problem because, as I mentioned, the effects data are also very different. Apples and pears here, as you can see on the slide. Now let me explain how we can align effect data. And I would like to do this with the following example. I already mentioned the effect mechanism of food dilution. Now just imagine that you could get sick from eating too many apples, right? Our stomachs have a limited volume, an apple has a volume. So yeah, getting sick from too many apples could mean that the total volume of the apples reaches a certain limit, namely the volume of your stomach. If that would actually be the effect mechanism for getting sick from eating apples, then yeah, it's probably just the volume that causes it, right? That was the basis for the food dilution effect. But then eating pears could basically cause the same effect, as long as the total volume of the pears would be the same. So the challenge then is to mathematically convert the volume equation for an apple to that of a pear. Now you see some equations here, and these actually are the equations that you would need to translate, for instance, from a sphere to a fragment, from a fiber to a fragment, or from all those individual shape categories to the mixed shape category distribution that we have for environmental microplastics that we see inside marine organisms. Now, this can be done, and this has been done. And then the aligned effect data can be combined with the aligned exposure data to get a consistent risk assessment. In short, the pipeline for the risk assessment methods that we have developed is that you start with literature search at the left-hand side. That's where your data come from. Exposure, but also effect data. Then you have to check whether the data fulfill the purpose of risk assessment, the QA/QC. The data that you then still have are aligned so that they all relate to the size range 1 to 5,000 micron. And then you can just compare and do a probabilistic risk assessment where if the risk characterization ratio would be higher than 1, you would say, well, there could be a risk. And if it's lower than 1, no risk. Now, this is a fairly binary approach. It is better to do this probabilistically, where this risk quotient is given as a distribution. Now, it takes too much time to explain that in detail, but I hope that the main idea is clear. Then the rest of my talk is just giving some examples, some applications. This is a study done by Gert Everaert. Gert Everaert Belgium. Here at the left-hand side, you see a so-called species sensitivity distribution. So this is for marine species. I realize now that it may be a bit difficult to see what's on the axis, so I will explain. It's a cumulative frequency distribution again. And on the x-axis, it's the effect thresholds for the different species. Now that means that the most left-hand side data point is the most sensitive species. And the most right-hand side data point is the least sensitive species in this list of species. And they're just ordered in the order of decreasing sensitivity, I should say. And then a curve is fitted. And from this curve, you can fit, for instance, the concentration of the x-axis, where only 5% of the species would be affected. And then you could say, well, let's use that as our more community-based effect threshold. So not a single species effect threshold, but an effect threshold which does at least some right to the fact that there are multiple species in a marine community. Now, this is done. This effect threshold is taken from this graph and then compared with exposure concentrations in the global oceans. And this is what you see at the right-hand side here. And it's done for different years. So 1970, everything seems still okay. 2010, then in the Mediterranean, you see this small red spot. So there are already some locations where the risk threshold is exceeded. Predicted for 2050, even worse. And in the year 2100, a huge risk, especially in the Mediterranean. But you also see a clear, reddish color in the North Pacific gyre, also explained by Tony. And also the Yellow Sea at the right-hand side of that world map shows a clear risk. Now, the last figure is here again, a bit bigger so that you can see it better. The darkest color here, so that would be the peak in the Mediterranean, is about between 70 and 184. That means that the exposure is between 70 and 180 times higher than the exposure concentration. Now, this is a prediction. What I always learn from this is there is still uncertainty. We would maybe not exactly know when the risk starts to occur, but it's quite clear that there will be a risk. It's more a question of when exactly than whether there will be a risk. It's interesting to, of course, talk a bit about why the Mediterranean. Well, that has to do that it's surrounded by densely populated countries, and that it's a basin which is fairly closed, right? You have Strait of Gibraltar of the west side, so it's not an open ocean. It's closed, so whatever is coming into this marine system, stays there for a longer time. And that's one of the reasons why the risk is quite high. Another example that's done later in paper from Scott Coffin in 2022. It uses a similar approach, and it's done for San Francisco Bay. Here you see a graph quite similar to the one I showed when I explained the alignment of the exposure data. It's cumulative frequency distribution, so it goes from 0 to 100% of the on the vertical axis, and on the horizontal axis, the particle concentration. And then the blue and yellow curves are for the wet and dry season, respectively. So there was a difference in the concentrations measured. And the yellow and blue curve are just the concentrations. So it's a cumulative frequency distribution of the exposure concentrations. And then the concentrations here are compared with 4 thresholds. So I explained about one threshold a couple of slides ago, to protect 95% of the species. So the concentration where 5% of the species might be affected. Now here in this study, 4 thresholds are used. And threshold 3 basically agrees more or less to 5% of the species being affected. That there are multiple thresholds relates to the fact that these thresholds were made in the context of a risk management plan. Risk management then has to do with well, if there's a certain level of risk, then what would be the appropriate management action? For the details, I would like to refer you to this paper. You can read all the detail there. But yeah, you can see that basically all the thresholds except for the last one are exceeded in San Francisco Bay. Now, and threshold 3 already meant a certain ecological risk in this bay. Another way of presenting the same data is in this map of San Francisco Bay, where you see certain locations. And please look at the legend. The green circle would mean that the exposure concentration is lower than the effect threshold. Now, you see that this is the case at the left-hand side in the Gulf of the Farallones, and that inside the bay, concentrations are quite a bit higher, and that the thresholds are exceeded. Now, the 4 thresholds have a different management implication. For instance, the first thresholds agrees to, then investigative monitoring is triggered. Second thresholds means that the discharges should be monitored. Third threshold means that management needs to be planned. So things have to be done to mitigate the problem. And exceeding the fourth threshold in this risk management system would imply that the sources should need to be controlled. And this comes from the same California regulatory exercise that Tony Walker also referred to in one of his slides. This brings me to the end of my presentation. I kept my take-home messages quite short. So I think the good news is that there are prospective risk assessment methods available. They come with uncertainty, but still they can be quite handy and important in answering the question of, so what? There are particles, but do they actually cause effects and also risks? And that we can use these tools also to make predictions, as I showed, until the year 2100. At the moment, the assessments that have been done already hint at the exceedance of risk thresholds in marine systems. And with that, thank you for your attention.
Wonderful. Thanks to you also, Bart. That's very interesting information. I see a couple of questions coming in as well, so please do send your questions. We'll come to those afterwards and pass those around to the panelists. Bart, if I could ask you to stop sharing your screen, that would be great. And then with that, I will pass over to Mehrnoosh. Please go ahead. Over to you. Thank you.
Thanks, Oliver. Hello, everyone. Good morning, good afternoon, good evening, wherever you are, and maybe even good morning. My name is Mehrnoosh Azodi. I'm a program management officer at the Secretariat of the Basel, Rotterdam, and the Stockholm Convention. For today, my presentation is a bit different from Professor Koelmans and Professor Walker. It addresses the policy side of nanomaterials and how the Basel Convention looks at nanomaterials and more specifically waste containing nanomaterials. In this presentation, we'll look at some key milestones and current work that is related to waste containing nanomaterials as we're preparing for the Basel Convention Conference of Parties that is scheduled to be held from 28th of April to 9th of May this year. So what is Basel Convention? It's a quick overview for people who may not be familiar. Basel Convention was adopted in 1989, and its overall goal is to promote environmentally sound management of hazardous waste and across the world. Today, it has 191 parties, and it looks at the hazardous waste and other waste from various aspects. One important part of it is control import and export of hazardous waste and other waste through a prior informed consent procedure. Basel Convention has grown in including different types of waste streams, including plastic waste that was included as a form of plastic waste amendments a few years ago came into effect. And probably you guys who are familiar with, you know that since then, a Basel Convention is the only international legal entity that oversees the trade of plastic waste and other waste. To give you a history of what happened to the waste containing nanomaterials, the Basel Convention Conference of Parties, in its 13th meeting, made a decision. That decision is on the screen, decision 13/17, and it took note of increasing presence of nanomaterials in waste streams. It requested the Secretariat of the Basel Convention to compile a report that would address issues related to waste containing nanomaterials and to also explore options for work under the convention. The report title is on the screen, and it's publicly available on our website. You're welcome to download it later. It covers a few topics and generally introduces nanomaterials, the waste containing nanomaterials, the disposal of waste containing nanomaterials, discusses some issues in terms of environmental health and to human health and to the environment, and also presents some existing activities that have been overtaken over the years to address the waste containing nanomaterials, and at the end proposes some options to further work under the Basel Convention. Coming to those options that was proposed in that report, it was proposed to classify the waste containing nanomaterials under the Basel Convention, provide information that is needed to develop the strategies for the environmental sound management of waste containing nanomaterials, minimize exposure to the nanomaterials during the handling and disposal of waste that contains those materials, and generally awareness raising and information exchange about the waste containing nanomaterials. In the next COP, the COP 14, the Conference of Parties in its 14th session, reviewed the report, this report that we talked about, and they made a decision, and that decision is 14/14. It took note of the report and recognized its findings, and then encouraged parties and others to do a few things, to, for instance, undertake further research and develop measures to generate information on this issue, encourage the parties and others to develop the strategies for environmentally sound management, or how we call it ESM, of waste containing nanomaterials, and also requested the parties to submit to the Secretariat any information that they have related to this topic, including case studies and best practices. So since then, we've continued to collect the submissions based on that decision in the COP 14. The parties and observers sent us their submissions and information, and that included the national studies, experiences with regulations and monitoring of nanomaterials in different countries, feedback on potential guidance, priorities, and case studies. As you can see, I put a screenshot of the Basel Convention webpage here on this slide, because this information, all of this that we have received from the parties and observers is online. You can access them by going to the Basel Convention website, to the waste containing nanomaterials, and from there to the best practices. There you can find all the submissions. And it's important to note that this information can help inform future deliberations and can serve as a knowledge base for countries who are beginning to address waste containing nanomaterials. The COP14 was not the last time that a decision was made on nanomaterials, and in the subsequent COP 15 and 16 to the Basel Convention, more decisions were made. And you can, again, on our website, go to the decisions tab and look at those decisions. Those decisions encourage the parties and others to continue submitting information to the Secretariat on existing guidance and regulations, best practices, and case studies. That brings us to COP17, the 17th meeting of the Conference of Bart parties to the Basel Convention. That is scheduled pretty much exactly in a month. And there's a document related to waste containing nanomaterials. That is document 17/11 that is publicly available on the, if you search Basel Convention's COP17 documents, you'll be able to find it. It proposes, signals the next opportunity for the parties to consider further work or decisions on this topic. And it proposes that the Conference of Parties may wish to adopt a decision along the following lines that you see on this screen. For instance, to take note of the information that so far is provided by parties and observers, which are online on our website, invite them to make available to the Secretariat any other new information by 31st of December this year, and then request the Secretariat to make any of those information that is made available to them up on our website and made available to everybody, to continue organize activities to raise awareness among parties and others on issues related to waste containing nanomaterials. And at the end, asks the Secretariat to report on all of this and implementation of this decision at the open-ended working group at its 15th meeting and the next Conference of Parties at its 18th meeting. To, as a result, to support awareness-raising activities, Basel Convention Secretariat has partnered with UNITAR, and we've been delivering a webinar series on nanomaterials and nanoplastics and microplastics. So far, we have had 3 of these webinars. The first one was a general one last year on sound management of waste containing nanomaterials. You see a screenshot of that on the right side. We had the first of webinars on macro nanoplastics on October 1st, and this is the second one, focused in the marine environment. We'll have more webinars throughout this year. Stay tuned. And if you have missed these 3 webinars, you can find a recording of those on the BRS website and also on UNITAR website. You can check them out later. Thank you very much. You can reach out to us with any information, case studies, articles that you have related to waste containing nanomaterials. My email is on the screen. And do check out our website. You can search Basel Convention waste containing nanomaterials and see what's available. Thank you. And back to you, Oliver.
Thank you very much, Mehrnoosh. Very good to see, and a reminder of though UNITAR's journey in terms of nanomaterials started under SICOM and now the GFC as an emerging policy issue. There are also very strong links to the waste containing nanomaterials under the Basel Convention, which we very much hope to continue into the future. So thank you very much to our panelists. As mentioned, we will do a question and answers session just while we've got a bit more time on this webinar. We've had one question come in the Q&A, and we have 2 in the chat. It mentions risk assessment. I know that certainly, Bart, you were talking about that, but of course, if others want to chip in, please do so. But perhaps, Bart, I'll invite you on this one. The question is it possible to assess potential risk associated with chemical releases from microplastics, or does the wide diversity in particles make that impossible? Thanks.
Yeah, Thanks for that question. It is true. I did not talk about the chemicals in the plastic to make life not too complicated. It is already complicated enough, but the answer is yes, there can be risks from release of chemicals from plastics, additives or plastic associated chemicals, whatever. Yeah, and basically it is the same principle. Also for chemicals, a lot of chemicals, effect thresholds are known. So in the case that you would know about the chemicals, or the chemical is known, or the mixture of chemicals is known, there are approaches to compare the exposure from the chemicals that leach from plastics to critical effect concentrations, and then draw conclusions on risks. There are plastic inclusive bioaccumulation models where the uptake of plastic particles is modeled and quantified, but also the quantity of plastics that would desorb from the plastic particles inside the body. These models are there. They are parameterized based on realistic data, realistic empirical data. So it's not modeling in the sense of completely hypothetical. These models are calibrated and validated. So the tools are available. There are two challenges, I would say. And that is one is about the nano-sized materials. So nanoparticles in general, and also nanoplastic, have a very high surface to volume ratio. So they can hold quite a bit more chemicals per unit of mass than bigger particles. The surface component absorption is bigger, especially for certain polymers that have a bit of a higher aromatic content or nature, like polystyrene, for instance. Now, these nanoparticles are difficult to measure in organisms, marine organisms, or whatever organism, but also we do not exactly know where they end up in the body. So the biodistribution of the particles is, that's still open territory. Let me put it like that, in a sense of research. And it also means that the exposure of chemicals via those very small particles is difficult to assess. The other big challenge is the huge number of chemicals. So we know a lot about a lot of chemicals, and there are read-across methods available, but still there are also many chemicals that we will not really know about whether they are there, and then it's difficult to assess the risk if the chemicals simply are not known. The research effort would be huge to find out which chemicals are present exactly in a certain environment in the oceans, for instance. I hope that answers the question.
Thank you very much. George, I noticed you had your hand up, but you've removed it. Do you want to jump in?
No, thank you. This was just by coincidence, yes.
Okay, all right, thank you. Mehrnoosh, I wondered if you wanted to jump in here. We have two questions about waste-containing nanomaterials. Do you see those in the Q&A?
Actually, I don't see them. Do you mind reading them?
Yes, so we have, please, I would like to know if there is a way to identify waste-containing nanomaterials, and is all waste-containing nanomaterials considered hazardous? So just thinking of the links there to the Basel Convention, but of course, if any of the other panelists would like to jump in, please do.
From the perspective of the Basel Convention, not all the waste that contains nanomaterials is hazardous. So Basel Convention right now does not include the nanomaterials or waste containing nanomaterials specifically under the convention requirements. So the decisions are really limited to gathering information. So that's the perspective of the Basel Convention on the issue right now. And then it's the first part of the question, do you mind repeating?
Is there a way to identify waste containing nanomaterials?
That's a very, very good question, but it's a broad question. I think it's really, you need to look at it case by case and the type of waste. There are techniques to identify and look into the nanomaterials, but again, it depends on the type of waste that we're looking at. For instance, sewage or wastewater, if we consider it as a type of a waste, there are techniques that you can look and identify and detect, and to a certain extent even quantify the certain types of nanomaterials in them. But again, I think this is a field that is evolving, and it really depends on what type of nanomaterials we're talking about, and again, what type of waste we're talking about.
Okay, thank you very much. We've just had an interesting one come in. I'll throw it open to any panelists who would like to offer. What is the main environmental source of micro or nanoplastics in human tissue? Bart, I see you've jumped in there. Please.
Yeah, I volunteered. Thank you for that question. Of course, the topic of the webinar is more like marine systems, but yeah, it's understandable that these very relevant questions also are being asked. So for humans, people mostly talk about three pathways: inhalation, ingestion, and a dermal pathway. And then it's quickly said that dermal is possible, but altogether would imply a fairly small flux compared to the other two. So then it's ingestion and inhalation. And well, where inhalation, the particles that we inhale come from, that's clear. That's from the air we inhale. Indoor concentrations are higher than outdoor. Ingestion, well, that's drinking water and food. And to a limited extent, some calculations have been done, assessments have been done, and they seem to suggest that the inhalation is a major source for entering the human body. So not like ingestion is negligible, but that inhalation is a bit more important. That cannot be one-to-one translated to the complete absorption by the particles by the body. So it's not really known how fast and well particles that are deposited in the lung tissue, the different parts of the lung, are being taken up systemically, so by the whole body. Whereas the same applies to ingestion. Then it passes the gut barriers and can be taken up in the circulatory system. So how fast and how systemic the uptake then is that's still a topic of investigation at this moment.
Thank you very much, Bart. And I note you also addressed slightly one of the previous questions about considering dermal exposure. So thank you very much for that. Tony, I see you want to compliment, I assume.
Yeah, I'll just build on that, because that is a $1 million question, isn't it? And like Bart, I would consider dermal possible, but much lower risk than the other two pathways. And I'm just going to provide some anecdotal evidence from a study conducted by colleagues in Portsmouth. They measured the concentrations of indoor air in a typical residential properties across the UK. And if you were to really think about prevention methods in your home, then that's still possible. And they found the highest concentrations of synthetic particles in the air, i.e. microplastics, atmospheric microplastics indoor, was in children's or infants' bedrooms. Because as you can imagine, they have a lot of plastic toys, synthetic fabrics, blankets, etc., etc. So if you did want to limit your exposure and limit the exposure of your family members, you might want to think about some of the furnishings and soft toys.
Thank you very much. A good addition for that more personal view of thinking on this. I saw Mehrnoosh answered one of the questions online, so thank you very much. We're coming towards the end, but there's also one question here we had, and again, I'll throw it open to the panelists. Does the degradation of microplastics influence the adsorption capacity and release dynamics of PFAS in varying aquatic environments? So if anyone has a view on that they could offer.
That's a difficult one. So We have done some experiments. It's still unpublished, but I can say something about it that show that the absorption of PFAS in aquatic systems to plastics is fairly low. Well, low if you compare it with trace metals, to what extent they have an affinity for the surface of microplastics, or even more, the affinity of hydrophobic chemicals to plastics. Plastic particles are passive samplers for hydrophobic chemicals, and they have huge partition coefficients from the water. For PFAS, it's much less. It's more like 1 to 300 liter per kilogram, whereas for a PCB it would be 10 to the power of 5 or 10 to the power of 6 or even 10 to the power of 7. So that's 4, 5, 6 orders of magnitude lower. So that is if the PFAS or the PFOS is environmental, right? If it somehow would be associated with a product, then I would not really know how the distribution would look like.
Okay, well, thank you for offering that. And then I'll do one last question, and then we'll pass over to George for his final comments. There was one we had on what health endpoints do we need to look into, both carcinogenic and non-carcinogenic? Go for it, please, Bart.
Yeah, so if the question is about the marine environment, so then we would need experiments done with marine organisms that specifically focus on cancer, or do not specifically focus on it, but they observe somehow still. And I'm not aware of those studies, so that doesn't mean anything. I can have missed some things. So maybe inflammation would be close in terms of the mechanism. And there are some studies that show it, but yeah, it's very difficult to make a causal link. Also because cancer may develop over longer time spans than those tests in the lab typically are done. So yeah, that makes it difficult to draw any firm conclusions. I know that there's interest in the same question more from the human health perspective. And as far as I know, this is being researched in various places. But yeah, I would not be aware of the best answer for that on this moment.
Okay, well, thank you for trying anyway. That's much appreciated. George, I see your hand up.
Yes, please. Actually, I think it depends very much which chemicals, additives are added in the plastic material. I think the plastic material per se is maybe not so much carcinogenic. At least the gastroenterologists, they use it huge amounts when you have to go to go to the gastroenterologist for making he uses his gastroscope, and then of course you have to empty your whole system before using polyethylene or similar plastics. But I think it depends very much on the what is in the plastic, and this makes the thing so complicated because there are thousands of chemicals or even more, and some of them could be carcinogenic. Thank you.
Thank you very much for adding to that. So thanks very much for the questions. I know we haven't been able to get to all of them. If any colleagues would like to perhaps type any answers if useful while we wrap up, please go ahead. Otherwise, I will thank all the participants, all the panelists, and pass over to George for some concluding remarks for us. Thank you, George.
Dear participants, my name is Georg KARLAGANIS, and I'm a senior advisor at UNITAR. First of all, I would like to echo Oliver and thank you for your participation in the webinar, and special thanks to our speakers, Tony Walker, Bart Koelmans, and Mehrnoosh Azodi for their great commitment and insightful presentations. They have invested a lot of time during day and night and energy in this event. First, we had a good overview from Professor Tony Walker of the microplastic pollution in the marine environment. He began with a historical overview of micro-nanoplastic research over the last 50 years. The current macro-micro-nano research comprises about 10,000 original peer-reviewed papers. Material flow analysis show that microplastic on the sea surface accounts for only 1% of the total amount of microplastic. Most of it accumulates on the seabed. Microplastic also enters our food chain in seaweed, mussels, oysters, lobsters, and commercial fish. The problem starts with littering. Littering means exposure of plastic in the environment. There are various strategies to reduce plastic pollution, including the ongoing negotiations for a global plastic treaty, the plastic waste amendments to the Basel Convention, various regional strategies and market-based instruments such as plastic-bagged levies and bottle deposits. Professor Bart Koelmans cooked for us a good pizza quattro stagioni with the 4 parts: risk characterization, effect assessment, exposure assessment, and risk communication and management. He got ecologically relevant metrics from food dilution and oxidative stress studies. He collected surface area and volume data of microplastic, also using machine learning modeling. He explained us the continuum of microplastic bioavailability. He faced several challenges during the quality control of literature data needed for risk assessment. His microplastic risk assessment pipeline consists of literature research, quality assessment, and quality control screener alignment methods, and probabilistic risk assessment. In this context, he looked also at affected species and effect thresholds. He has tools to quantify risks for global surface waters, large lake ecosystems, sediments, and global oceans. As an example, he showed risk assessment data of San Francisco Bay. His take-home message is the following: prospective risk assessment methods are available. They hint at the exceedance of risk thresholds in marine systems. Dr. Mehrnoosh Azodi gave a short overview of the work on the waste-containing nanomaterials under the Basel Conventions since COP13, under the upcoming COP17, which will take place between 28 April and 9th May 2025 in Geneva. The Secretariat, as always, welcomes parties and observers to the Basel Convention, but also all relevant stakeholders to reach out and share information and latest updates regarding the sound management of waste-containing nanomaterials. Now, we have a survey to seek your feedback, and through this, we kindly request you to let us know if this webinar was useful and if you would like to have more webinars on related topics. Once again, many thanks to the speakers and thank you for your attention. Over to you, Oliver.
Wonderful. Thank you very much, George. I also see some suggestions for perhaps a next webinar, next focus of topic. So that's gratefully received. That's also the kind of information we're looking for from the survey. Some ideas. We do want to continue this series we've developed. We want it to be useful to participants, stakeholders of different sectors, different regions, different stakeholder groups such as government or academics or otherwise. So please do fill in that survey. Please let us know what you think, and we will try and satisfy those suggestions and requests in the future. But from my side, thank you very much. Thanks to our panelists for very willingly sharing their information, sharing some of their extra knowledge as well to answer the questions as we went along. Thanks to my colleagues Georg and Mehrnoosh who've helped set this up, and Jiang in the UNITAR team. Again, a pleasure to do this webinar with you. We will share as much information as we can on our webpage, so I think that's okay already with our presenters for the presentations, and we will put the recording up as well, and hope to continue sharing such information in the future. So we have the QR code for that survey. It's also been posted in the webinar chat for you. But from that, I will say thank you very much. I'll close there and hope to engage with as many of you as possible in the future. Thank you very much. Have a good day, everyone.
Bye-bye.
Goodbye. Thank you very much.
Thank you, everyone. Toodaloo.
Thank you. Bye.